Hello, everyone. Thank you all for joining us today for this Lunch and Learn panel to explore roadblocks that can occur during the process of developing gene and cell therapy treatments. My name is Emily McGinnis, and I am the Chief Patient and External Affairs Officer at Taysha Gene Therapies. I am also a member of the ASGCT Patient Outreach Committee. Today, we have the great pleasure of hearing from industry, clinical, and FDA perspectives on the various challenges and roadblocks in the development of gene and cell therapies and opportunities to address them. Please note, we will not be able to discuss specific disease states or products during this panel. Today, we have a great panel, as mentioned earlier, and we'll go through and ask everyone to introduce themselves, starting with Dr. Kimberly Goodspeed, the Assistant Professor of Pediatrics, Division of Neurology at UT Southwestern Medical Center. Kim, can you say a few words about yourself?
Sure. Thanks for the introduction, Emily. So I'm Kim Goodspeed. I'm a child neurologist in Dallas, and I work really closely with our gene therapy program that's been growing at UT Southwestern. So my specialty is in neurodevelopmental disorders, so taking care of kids and families with autism and intellectual disability, but I work super close with our non-clinical research labs as well as our clinical folk who are trying to get things ready and moved into clinic. I'm excited to be here and chat through everything.
Great. Thanks, Kim. Our next panelist is Dr. Jennifer Helfer, who's a Senior Director of Patient Advocacy Engagement and Corporate Affairs at Viridian Therapeutics. Hi, Jennifer.
Hey, hi all. I'm honored to be here and part of this awesome panel. As mentioned, my name is Jen Helfer, and I'm serving today on the panel as our industry rep. I am a member of ASGCT's Patient Outreach Committee. As for employment, as mentioned, I work at Viridian Therapeutics. I would like to note that this is not a gene and cell therapy company. However, I have worked at 2 different gene therapy companies, most recently a little over a month ago, and I've also consulted for a handful of gene and cell therapy companies. With that, I would just like to mention that all views I express today are solely mine and not necessarily reflecting those of my current or past employers. Thank you.
Thanks, Jennifer. Next, we have Dr. Jajashree Purohit-Sheth,
Hi, good
acting
afternoon.
director. Good afternoon. Nice to see you. At the Office of Clinical Evaluation and Office of Therapeutic Products at CBER at FDA.
Thank you, Emily, and thank you for the invitation to participate in this panel. As Emily mentioned, I am the acting director of Office of Clinical Evaluation in our newly formed Office of Therapeutic Products at FDA. I've actually been at FDA over 20 years now. I have experience, about 10 years of experience in CDER, the Center for Drug Evaluation Research, about 5 years in the Center for Devices and Radiological Health, and over 6 to 6.5 years now in CBER in the Therapeutics Products program. So I am happy to be here and participate in this panel discussion with you today.
Great. Thank you, Jajashree. Our next panelist is Dr. Sandhya Sandhuja, who is the acting branch chief of the Office of Pharmacology and Toxicology with the U.S. Food and Drug Administration. Sandhya, I'd love to hear more about your background.
Hi, Emily. Thank you. Good afternoon, everybody. I'm Sandhya Sandhuja, and I'm acting branch chief for Pharmacology Toxicology Branch 2 in our newly formed super office, Office of Therapeutic Products, which was previously known as OTAT, Office of Tissues and Advanced Therapies. I joined the FDA in 2016 as a joint NCI, NIH, and FDA fellow, and I worked briefly with CMC for a year before I moved to Pharm Talks in 2017. Prior to joining the FDA, I did my postdoctoral research at the Whitehead Institute of MIT. It's a pleasure to be here today.
Thank you to our great panelists for being here. I'm really looking forward to the discussion. As we mentioned, we're going to talk about various roadblocks on the way to developing a cell and gene therapy treatment. And in order to set the stage, Jennifer's going to do an overview of the clinical trial process. So Jennifer, if you can walk us through this, that would be great.
Yeah, happy to. So as you can see here on the slide, this is a quick overview of the drug development process for gene and cell therapies. More information can be found on the ASGCT's website, so I definitely recommend you check that out after today's webinar. So to briefly go through this, before we can prescribe, or a healthcare provider can prescribe a cell or gene therapy, we first need to show through clinical studies that this therapy is both safe and effective, and that the potential benefits outweigh the possible risks. To do this, it can take many different types of studies that last years or even decades, and it all starts back in the laboratory. So these early preclinical studies start by developing and testing multiple potential gene therapy candidates. The most promising of these then continue on for more preclinical testing, sometimes in models of specific conditions. And this involves gathering information on how much to give, so the dose, how to give it, so the route of administration, and looking at in these models what are the benefits we're seeing and what are some maybe of the side effects and safety risks that we might see. These preclinical studies help refine the potential gene therapy itself, and then also prepare it for continuing on into clinical studies with people. Now, to start these clinical studies, a sponsor first needs to get permission from country or regional regulatory agencies, as well as local ethics committees. Regulatory agencies like the FDA and local ethics committees, they decide if this therapy is safe and ready to move forward into clinical studies. And they do this by reviewing everything, all of the data and everything that was done in that preclinical stage, as well as how is the therapy manufactured and so much more. Once the sponsor is given the green light, early clinical studies start. Traditionally, these are separated into phases, into 1, 2, 3, and 4. With gene therapy, though, many times phase 1 and 2, so these early phases, they're combined. And each of these phases is designed to answer a different question about the gene or cell therapy. Phase 1, 2 trials or studies typically look at what is the best dose to deliver the greatest benefit while also ensuring safety. And then also, is there enough information to support continuing on to adding additional participants? And that's usually then done in a phase 3 study. And here is where sponsors are really looking at the possible benefits of this therapy, and are they outweighing the risks? Once the clinical studies are completed, and there's enough data to support, that there is benefit here with this therapy, the sponsor goes back to the regulatory agencies, who do a very rigorous process and evaluation of all of the data. They're looking again at all the preclinical data. They then look at all the data collected in the clinical studies. As well as they talk to other stakeholders. And this is, the healthcare community, the patient community, and take all of that into consideration. Knock on wood, the therapy is approved. It is then a commercial product, but that's really not the end of it. There can be phase 4 studies where we then go and try to learn more information about gene therapies, as well as there's the access piece. And that means that after gene therapy is approved by regulatory agencies, we still need health insurance companies and national payer systems to work out pricing and reimbursement. Because without that last step, we still won't be able to provide access to these therapies. I could keep going, but again, go back to the ASGCT's website to learn more about the process. But I'm really excited to have a panel discussion about all of the different, roadblocks and hiccups that might happen along this journey.
Thank you, Jennifer. And just for the audience's awareness, we are going to walk through each of these sections during our discussion today. So we're going to start our discussion in the preclinical, or also known as the non-clinical section of this drug development process, especially for gene and cell therapy. So I'll kick it off with Sandhya. Can you tell us what types of non-clinical studies FDA looks for when evaluating potential gene and cell therapy candidates for an IND?
Yes, absolutely. Sure. And thanks, Jennifer, so for setting the stage with giving an overview for clinical development and also touching upon preclinical studies. So, before I answer this question, I just want to reiterate for the audience that what we call as gene therapies are products which mediate their effects by transcription or translation of transferred genetic material. And these gene therapies can comprise of conventional viral or non-viral vector-based products. They can be cell-based products which have been genetically modified, or these could be products incorporating genome editing. So, from a non clinical perspective, we are looking for well designed and well executed non clinical studies that have appropriate controls that can generate robust and interpretable data to inform safety and rationale for administration of the investigational GD product. We are looking for those data to support GD administration or gene therapy administration at the planned clinical dose levels via the intended clinical route of administration in the intended patient population. So there are these different aspects that need to be addressed and supported by data that are generated from these non-clinical studies. In addition to talking about safety, one other important aspect for non clinical development of gene therapy products is establishing proof of concept. And that is important to support rationale for gene therapy administration. It's a very important consideration, and there is a reason for that. These gene therapies, they come with an inherent risk. These are novel, much novel products that we have, seen being developed with novel technologies, with novel concepts. And that's why there is that inherent risk which could be associated with the actual product, the vector, the transgene. It could be associated with the delivery device or the procedure for administration. It could be a challenging anatomic site or route of administration, or an invasive procedure for delivery. So there, and in addition to all this, there may be some unknown long term risk that we just don't know. And that's why it's very important to conduct these non clinical studies to establish a proof of concept using animal models of disease or other available resources to provide that evidence of biological activity, and thereby supporting a favorable benefit risk profile for the product in that intended clinical population. I would also like to highlight here that for diseases that impact children, it is required by the regs to provide or demonstrate data supporting prospect of direct benefit. So if you're planning to initiate a trial in children, then you are required to provide evidence of activity to support prospect of direct benefit. And these data can come from your non-clinical studies in appropriate models of disease, showing that administration of your gene therapy product does lead to improvement of some disease relevant parameters to support administration in children. And these data can actually very well come from clinical studies in adults as well. But ultimately, coming back to the question with respect to non-clinical studies that should be conducted to support safety as well as demonstrate plausibility and establish proof of concept, it is important to take totality of data into consideration. So that weight of evidence approach to set up a favorable benefit risk is a key point from these non-clinical studies.
Thank you, Sandy. Can you add and talk a little bit more? That's a really, really great background on the robustness, really, of the package that's needed to evaluate these therapies. What are potential roadblocks during this stage of development?
Yes, that's a great question. And as I just discussed, how these non-clinical studies are, key to establish safety and rationale, and these data are required to support, moving forward into clinical investigation. If so the biggest roadblock is if such studies have not been conducted, or information from studies that have been conducted have not been provided in an IND submission as complete study reports. That's a big roadblock right up front to a successful IND submission. right? And then, additional roadblocks, of course, if non-clinical studies have been conducted, but the in general, it is decided, it is determined that these studies are not adequate to support safety and rationale for your proposed clinical trial plan. And there can be several scenarios where such studies could be considered inadequate, and for one or more reasons. For example, the product that was used in your definitive non-clinical studies is very different from your intended clinical product, and that's a big roadblock. The choice of animal model, disease model, or the species that was used to conduct these definitive study is really not relevant to test the safety or efficacy of the product. The product is not active, the vector doesn't transduce that cells in that animal species. So that's, these are some of the key deficiencies that can present themselves as roadblock to successful translation of non-clinical data to support clinical administration. The route of administration could be irrelevant, the one that's used in non-clinical studies. The dose levels may be not covering the intended clinical dose level. The delivery procedures may be significantly different. The duration of the study it's probably too short. If it's too short to capture, any delayed or potential late onset toxicities, that's a big red flag. If appropriate controls are missing, then it's hard to really interpret what these findings mean. If animal numbers are inadequate, appropriate intervals are have not been assessed for the different safety parameters, and if key safety assessments are missing. For example, if immune response hasn't been captured, or biodistribution in target as well as off target tissues hasn't been adequately captured, tumorigenicity is a concern which hasn't been adequately captured. And then, tying it back to one of the points that I mentioned earlier with respect to a clinical trial in pediatric population, if you really haven't identified a minimal effective dose from your non-clinical studies to support prospect of direct benefit for a clinical trial in pediatric population, that's a huge roadblock right there. So, these are some of the roadblocks that can be in during your non-clinical development. And I also want to, close this particular question by saying that, even if a sponsor may think they have conducted appropriate studies, their interpretation of findings and translation of animal data to humans should also be appropriately justified. If, say, safety findings have been seen in animals but they were not well characterized or well investigated to really inform what the risk could be in the patient population, that's a big concern. That could be a major roadblock. So overall, I think it's very important to understand that non-clinical data are key to support safety and rationale for a gene therapy product administration, and our approach in general is science and data driven with a lot of weight of evidence consideration. I also want to quickly mention that all what I have mentioned and touched upon, I would like the audience to also refer to the guidance document that are available on our website. So we have the preclinical 2013 preclinical guidance for cell and gene therapy products. We have several other guidance documents from recent years for gene therapy products for rare diseases, for hemophilia, neurological indications, and we have draft guidance for CAR T cell products and genome editing products.
Thank you. That's really helpful. I think it's important to note if you can share ways that sponsors can work with the FDA during this really important time period to make sure that they're doing all the studies. Can you share a little bit about that, Cynthia?
Yes, absolutely. And great. Thanks for projecting that slide. That actually is a great snapshot of opportunities that we offer to stakeholders, to our sponsors of our developers of gene therapy products to interact early during their development process. And these early interactions, which as you see are highlighted by that blue circle, are the interact and pre-IND meetings. Both of these opportunities are non-binding interactions for us to provide feedback for your non-clinical product development. Um, just to mention about interacts, these are, as the name says, these are meant for initial targeted discussion during early phase development when you have identified your gene therapy products and have conducted preliminary studies to evaluate proof of concept, and maybe have some data to evaluate safety, but definitely not when you're ready to conduct any definitive safety study. So you're basically from an interact meeting is meant to provide feedback on design of your more definitive proof of concept studies and product manufacturing plan. Going further, a next step further is a pre-IND meeting. So these meetings are meant for discussion of your definitive plans for safety assessment, your product manufacturing, as well as design of your actual clinical trial with respect to patient population, dose, regimen, eligibility, all those aspects. For both of these interactions, the sponsors should be submitting in their meeting package detailed summaries of their development status and studies that have already been conducted, their product manufacturing and clinical plans, and have specific questions for which they are seeking feedback. For more information regarding process and timelines for these meetings, all that is available on CBER's website. So I strongly recommend folks to visit our website for more details.
Thank you so much. This is really great overview. Thank you. And shifting gears a little bit, Kim, can you talk a little bit, given that we have advocacy groups and patients and families on the line, how can they work with either a sponsor or to overcome some of these roadblocks during this period?
Yeah, absolutely. So, what I think is really exciting and very cool is that so many of these programs actually originate from the advocacy groups pushing them forward. So much of that initial funding is from grassroots funding from the advocacy groups that they get those initial proof of concept studies off the ground. They may not be able to take it all the way to IND, but they get it started and they're really that catalyst. And so, often they can reach out to an academic physician, somebody like me or somebody in the lab, who can start some of those initial studies and maybe start developing the animal models if they don't exist. And it takes so much discussion and collaboration between the groups to be able to get those animal models right or the cell line models right, make sure that we're testing the right things, identifying the right targets, and really understanding what the patients look like. So it's this really team effort that often originates from the advocacy groups, kind of shouting their needs and getting people excited and involved, telling their stories, telling their family stories of the people in their groups, and being able to really help design those early studies.
Thanks, Kim. Is there, before we move on to the clinical section, does any other panelists want to add anything here?
I'd like to add to what Kim was saying. I think also another piece where advocacy groups can really play a part in this early phase is educating other stakeholders too, including the FDA and researchers about the disease, and kind of gearing up for that, clinical trials to start. So it's also educating the community about what are clinical trials, why are they important, why are they so long, why do we, all of the different questions that come along once the clinical trials start. If you start that education early, sometimes it helps the process down the road.
And I'll also add here, following on what Jennifer said, that during this early phase, sponsors should consider, or folks that are studying these very rare disorders, potentially doing some natural history studies to get some information regarding, what is clinically meaningful to the patient. So that may help in early phase, the IND study, as well as later phase studies, to identify clinically meaningful endpoints that can be evaluated and assessed. So the voice of the patient is absolutely important in identifying what would be meaningful to a patient. we can look at information, we can understand the disease as physicians and as product developers. And sometimes we may look at something and say, oh, well, we don't think this is significant. But for a patient, maybe walking from their bed to the bathroom, maybe just walking, 10 feet is clinically meaningful to them if they have end-stage COPD, or just walking outside on their porch for a few minutes. So what is clinically meaningful to a patient in the context of what they live with on a day-to-day basis. So if at least during these early phases, maybe doing some interviews with patients to understand what can potentially inform later clinical trial research and what endpoints we assess. So I think that is absolutely very important as well.
That's great. Thank you. I like to say, and you're right, that patients and families are experts in these diseases, not
Exactly.
more than anyone. Appreciate that. So shifting gears, when we kind of started going in this direction, is really moving forward with a clinical trial. And so, Kim, can you provide an overview of really what is the purpose of a clinical trial?
Yeah, so I think you'll hear us sound a bit like a broken record today, but I think as a physician educator, it's also repetition is important. That's how we learn. But safety and efficacy, that's really the heart and soul of a clinical trial. You can't give a new treatment of any kind to a patient unless you know it's not going to cause them harm and potentially, especially in the pediatric disease space, is going to potentially help them, potentially make their life better. And so that's really the goal of these clinical trials. We try to set up for safety and efficacy using those non-clinical studies to guide the design, using our patient advocates to help us identify what are those outcome measures or the things that we can assess or measure over time for improvement, but also a whole battery of safety assessments that may seem like a huge burden and lots of testing, especially in the early clinical trial phase 1-2 studies, but they're really meant to cast a really broad net to capture any potential adverse event or safety event that could come up in a clinical trial. And how do we pick those efficacy measures is really dependent upon the patient community. And so we will partner with them over time, especially in the non-clinical phase, trying to conduct some of those natural history studies that Tajari was mentioning. And those can be in the phase of a registry study where you just send out some surveys to your patient population. They can be relatively cheap to do and can get you a quick and dirty answer to what are some of those key metrics that you need to measure. But then you may want a prospective natural history study, where you take patients over time and they go to a couple different centers across the globe to be monitored in a standardized way. Do you need to develop a new measure because nothing out there is working right now? That's going to take a lot more time and effort. And so all of this goes into that planning phase and sets you up for success potentially.
That's great. Thank you, Kim. Jennifer, what considerations can be made prior to the study design of a clinical trial?
Yeah, I think to Kim's point, we're going to kind of repeat some stuff. And I think some of this was mentioned during, the previous conversation on the preclinical side. But I think it's important that a lot of considerations for the clinical trials and the designs actually happen in parallel to that preclinical research that's happening. And a lot of this that's done in parallel is talking to people. It's talking to the healthcare providers. It's talking to the patient advocacy organizations. It's talking directly to patients to really better understand what does current care look like? What does the network look like? What is going to be meaningful if a new therapy were to come out for these individuals? And then we have to figure out how do we then create a tool to measure that meaningful change and define our endpoint. And then we start thinking about, okay, we have endpoints. Are they primary, secondary, exploratory? There's those considerations. There's also identifying the right people for these trials. Unfortunately, gene therapy clinical trials or any clinical trial can't enroll everyone that lives with that condition. And so it's really thinking about who is most appropriate and who can benefit from the therapy from what we understand about it thus far. There's also other considerations about refining the route of administration. You have to start thinking about trial sites, who are going to be the principal investigators running these studies? Are they ready for gene therapy? Is the site set up to do that? How are we going to ship these therapies and one, manufacture them, but then ship them and get them to the sites, store them appropriately? There's a lot that goes into consideration, both in parallel to preclinical research as well as to the design of the clinical study.
Thank you, Jennifer. I think one thing that's unique about clinical trials and gene therapies is that trials are started in humans. And so, to Joshua, can you speak a little bit to why we start in affected individuals in gene therapies?
Sure, sure. So, in contrast to our small molecule drug products, many classes that are very well studied and established, there's relatively a lot less information regarding the long-term effects of cell and gene therapies. And cell and gene therapies, another factor compared to our small molecule drug products, they can persist in humans for extended periods of time following administration and perhaps have extended or permanent effects, even if the product is no longer present. So these considerations and risks to really subjects for cell and gene therapy is of utmost consideration in our mind. And so we don't really do healthy volunteer studies because of many of these risks, and a lot of this is evolving. We're still learning. It is, many will say it's still in its infancy compared to these very well established products that are on the market, these small molecule drug products. And so it really does not present a favorable benefit risk to enroll healthy volunteers. And so for all of these subjects that enroll in these cell and gene therapies, we start with the patient population. And another reason, in addition to risk, is for very rare disorders, we typically request that the sponsor evaluate some measure of effectiveness of the product. If you have a disease in, maybe just hundreds of patients, or maybe 3, 4, 500, or maybe a few thousand, it may be very challenging to enroll subjects in a clinical trial when the disease is very rare. So if this first study is assessing clinically meaningful measures of benefit, that may be the only study that a manufacturer may submit in support of a marketing application. So not only are we concerned about risk to healthy volunteers, but we also want to evaluate some degree of efficacy given the risk of these products in the intended patient population. So for most cell and gene therapy, we are looking at the benefit risk profile, and we do not believe that the benefit risk would be acceptable for healthy volunteers.
Thank you. Appreciate that explanation. Given the theme of this talk, Jennifer and Kim, can you talk a little bit about the roadblocks to enrolling a clinical trial, especially a gene therapy clinical trial?
I can jump in first, Jen.
Yeah, go ahead.
Sorry. So I think touching on what was just mentioned, I think the rarity of disease, often these gene therapy studies are designed for rare disease patient populations. That means that you don't have a whole lot of patients to not pick from, but not a very large patient pool in order to enroll from. And they may be international patients. They may be spread all across the globe and not conveniently centered around academic centers where these studies are going to take place. So I think that's your first big hurdle, is figuring out where are our patients and what are the burdens that they are going to face in enrolling in a trial. And really trying to take that family perspective of, a lot of these trials will require you to be local to the clinical trial site for several months right after injection or right after treatment. And you may also have to be traveling back and forth for years, and then continuing to follow up at least remotely, but probably coming on site for 5, 10, 15 years after you've been dosed. So what we're asking of families is no small feat. And it's going to take a lot of coordination on the family's part to be able to actually participate. And so I think that's one of our very first big roadblocks, is making sure that we have thought through all of those things. Do patients from international flying from international sites need a visa to come? Do they need health insurance while they're there? Do they need travel support? Are they going to have stable housing if they're going to have to stay long term? And how are we going to accommodate their loss of wages and loss of work? So all of these things really have to be factored in and considered when you're thinking about the logistics of actually starting a trial. And then how do you find the trial? So, how do you get the word out to your community? How does your community kind of talk with their local treating physicians about their own individual risks and potential benefits? And how can kind of families become that self advocate in both directions of educating their communities, but also educating their physicians when there's maybe a power hierarchy there that may be uncomfortable, but you're going to have to be that advocate and educating and sometimes even bringing the clinical trial to your physician because they may not know that it exists and is happening.
That was a perfect answer, Kim. I don't really have much else to add to it.
So I would just like to point out, Kim mentioned foreign sites being engaged or having subjects with rare disorders, and you don't have that many patients to go around, and they may have to come to the United States. But another roadblock that we've also seen is that for some of these INDs, there are global sites, so patients don't have to travel to the United States. They can be enrolled in their country or a country that's nearby. But sometimes for certain conditions, maybe in rare tumors or the oncology space, standard of care is something that is really very important. And a potential roadblock may be that you're designing a clinical trial, but the standard of care is different. And so you may be looking at a patient population that is relapsed or refractory to maybe 3 or 4 prior lines of therapy. And in the United States, for that particular trial, we have an understanding of what the standard of care is for our patient population here. And we're looking, honestly, to market this product in the United States, to have it marketed here. So we're going to be evaluating prior therapies in the context of standard of care here. So a roadblock that we have potentially, or we have seen, particularly in the oncology space, is that some of these foreign sites, their standard of care may be different. The chemotherapeutic agents they may utilize, et cetera. So enrolling subjects from overseas may be a potential roadblock because the standard of care may be different.
That's a really great point. Thank you for bringing that up.
Yeah.
And then another roadblock, and we talked a lot about this in the non-clinical section, was safety. Sandia, can you talk about some of the potential safety issues that may come up during this clinical trial timeframe that may be a roadblock in advancing to next steps?
Sure, yeah. So during clinical development, as your IND is progressing, your development is progressing. There can be scenarios where some safety signals may arise, or there may be scenario where you are making a significant manufacturing change to your product, and those manufacturing change may elicit some concerns associated with safety. And the point is, those manufacturing changes may be significant to really reevaluate safety of your product. So in general, those changes may trigger comparability studies at during your clinical development. But also in those comparability studies, there may be consideration towards bridging and non-clinical bridging studies to basically show that your changes in your product are now not, they really do not impact safety in animals, and non-clinical studies geared towards addressing that and evaluating that to see, okay, now this new so-called improved or changed product is still as safe and as effective as the prior version. if you may. And additional scenarios may also include where during clinical development a safety signal is identified, which was probably not seen or not informed by non-clinical studies, and now you have this safety signal. um, how can we really better understand that safety signal? Can we really do can we design new non-clinical studies now that we have identified this signal? Can we really identify an appropriate animal model or species to understand what could have caused this safety signal to arise? And really, how well can we understand it now that we see that safety signal to come up?
Thank you. Kim, do you have anything to add here?
Yeah, sure. I think one of the big things we think about in cell and gene therapies are immune responses. And so, just to dive a little bit more granularly on these safety signals that Cynthia is mentioning, is oftentimes these are in the realm of an immune response. So the body is reacting to whatever we have just put into it. As we mentioned earlier, a lot of these are virally mediated. So we're using a virus to deliver the therapy, and our immune systems are built to fight against viruses and fight against anything foreign. And so a lot of these safety signals come from that just inflammation that can happen in the body. Most of our protocols will also use some sort of medication or a combination of medications to reduce that immune response. So you may also have patients that are relatively immunocompromised. So their immune system is kind of dampened down or turned off a little bit in order for it to accept that new therapy. So you can have these reactions in kind of both directions, where you may have too much immune activity, but you may not have enough and be susceptible to other infections. So we've seen it go both ways in different trials. And so that's partly why we have such a burden ridden protocol with those phase 1-2 studies, especially to make sure that we're surveying the entire body system to see if any changes are happening that could show organ damage, so liver damage, kidney damage, heart damage, things like that. Also watching patients for infections. But it doesn't stop with that phase 1-2 study. We keep monitoring patients, right? We continue to collect data over as long of a period as we possibly can with our patients to make sure that they're staying safe and that we can really understand those long-term effects as well. Because as we've stated already, we're in the early days of gene therapy going into humans, and so we still have a lot to learn.
Thank you. Another roadblock that comes up is, unfortunately, there are either program halts or full terminations of programs. Jennifer, do you want to start by talking a little bit about that? Like, what's the difference, and how does this impact the patient population and even companies and yeah.
Yeah, happy to. No one likes when something gets halted or terminated. It's an unfortunate reality, though, of drug development. It happens both in gene and cell therapies, but also other types of drug development. So this isn't unique to this field at all. So I think halt, usually when I hear halt, I think hold, and it's a pause. And we hear clinical hold quite a bit. Termination is more around ending a program. And there's many reasons that these can happen. For clinical holds or halts, I think, we've seen quite a bit of these over the last few years, and they're typically due because of we've seen some type of adverse event, whether it's drug related or not. And a lot of times we're not sure. And so we have to, stop, pause, and figure that out and answer that question. I also can recall there was a clinical hold maybe a few years ago now that was related to actually transportability and the stability of the gene therapy to the clinical trial site. So it doesn't always have to be related to an adverse event. And then just, it does take time again to figure these out, figure out the reasons, work through them, and then move forward. For termination, ending a program, this, again, many reasons this can happen. This can be due because, early during the phase 1-2 the primary endpoint wasn't met. There was that proof of concept, but in the clinical study of meeting that endpoint, are we seeing an efficacy here? Wasn't there, at least determined by what we had specified. I think the other big thing and really unfortunate we're seeing right now happening is a lack of resources. It is really, terminating some of these programs. Many gene therapy companies are small biotechs. They're not commercial entities, so they don't have streams of income. And so there's a lot of resources and money that goes into developing these therapies. And so sometimes you have to make that hard decision to terminate a program due to lack of resources.
Sure. Tejasri, is there anything you wanted to add?
Sure.
There?
So from, the FDA's perspective, Jennifer mentioned clinical holds, right? So we typically, many of our holds are with the initial IND submission, and it's because we may not, for many of these, we do not have sufficient information to assess risk to subjects. That's probably one of the biggest reasons that we put this on hold. A sponsor may believe they have everything they need. When we evaluate it, we may not have adequate product characterization, for example, that we would require to allow dosing in humans, or we may not have adequate preclinical data. And Cynthia touched upon this, considerably in the earlier portion of this panel discussion, so I won't reiterate here. So we may not have adequate information to support the dosing or the dosing regimen or the route of administration. And so we will put the trial on, clinical hold, but it doesn't mean that it's terminated. We say, please come back. We provide extensive information in our hold letters to the sponsors, so they're aware of what we would require from the sponsors to address our clinical hold concerns and issues. We also may put a trial on hold if there are safety signals that are identified during the conduct of the clinical study. And so when we evaluate, we look at, we evaluate, okay, what was this? And first we want to understand, is this something that we think is related to the product? And it may not just be the product, it may be the delivery of that product. Many times we have intracranial administration of products for neurologic conditions. And sometimes the procedure is risky, and you may have significant adverse events due to just the procedure itself and not necessarily. But when we evaluate, we do not evaluate the risks of the investigational gene therapy, for example, in isolation of the delivery of that product. So if we identify a significant safety issue, we may place the IND on hold at that point as well. And then, Jennifer had mentioned that, why would a program be terminated? Well, yeah, if you do your phase 2, phase 3 trials and you don't identify that the product is effective. But, we have also seen rarely that programs have been terminated because over time during the course of the initial clinical trial, there are unreasonable risks to the study participants. And often we can mitigate risk if we know what it is. We can incorporate risk mitigations in the clinical trial protocol, revise the protocol. But there may be certain risks that we just cannot mitigate. And if you see them in majority of the subjects with long-term clinical sequelae, morbidity, disability, we may put this trial on hold, and the sponsor may not be able to address the significant safety concerns that we have, and they may decide from a safety perspective to also terminate the program.
And how can, and I'll put this to you, Jennifer, how can patients and advocacy groups help overcome some of these roadblocks we've talked about today?
Yeah, I think, one is, they can work with the sponsors to really help the community understand what's going on. Why is this on hold? What does this mean? What does this mean both for people who maybe are participating in the studies, for people who are waiting to be screened for the studies, and just to help that communication and keep the community informed, because it does sometimes these holds take a while to get off of hold and have that hold lifted. So I think there's a constant communication and education that needs to happen, and it's best to come from the advocacy groups during this time. I think, also if there are opportunities where the advocacy groups, can help too, working with HCPs or other stakeholders, it takes a community to bring gene therapies to the market. And so working together is the best way for us to overcome any roadblock.
And to Jessary, can you speak to ways that patients and families and advocacy groups can interact with the FDA? I know there's been lots of opportunities for that created.
Sure. And so, FDA has been very interested in patient engagement opportunities over the last several years. We have patient listening sessions. We will host a series of these, and we have held these, where we have a select group of patients and caregivers. It's a maybe sometimes 10, 15 individuals, but we have questions for these patients and caregivers about the particular disease. What is it like to live with the disease? Not only from the patient perspective, but also from the caregiver perspective. And then to understand what their most urgent needs are with respect to this condition. We also like to hear from our patients and caregivers about what they would consider a clinically meaningful benefit in this condition. So we have been engaging in these patient listening sessions through a collaborative effort with the National Organization for Rare Disorders and the Reagan-Udall Foundation for the FDA. We also have a patient engagement collaborative. This is a patient group established by the FDA and the Clinical Trials Transformation Initiative. And this PEC is composed of individuals and patient organization representatives who discuss topics focused on how we can enhance patient engagement in the development of medical products and engage in some regulatory discussions with FDA. So those are two avenues. We also have the FDA Patient Representative Program. And this is one of the agency's primary mechanisms for recruiting patients and caregivers who have experience with the disease, condition, or medical device. And they may participate as special government employees. So, for example, you may be familiar with our product approval Luxturna for a rare retinal disorder. Now, this is a disorder for which we had no available therapy, so there was really no pathway that we could leverage from a prior approval. So we engaged a patient representative to provide their feedback in the context of that product development program to identify what would be clinically meaningful. So that we found very, very valuable. So this was done in the context, first as a product was being developed, to get some idea from a patient. And then also in the context of when the marketing application came in, we have opportunities to request a special government employee to address some questions that we may have in the context of risk benefit. And then we also have the patient-focused drug development initiative. Initially, FDA led series of 24 of these patient-focused drug development meetings geared towards a variety of different conditions and disorders to better understand the patient's experiences, perspectives, their needs, and what their priorities are, and to have this captured in a meaningful manner. We have a lot of these patient-focused drug development meeting summaries that have been posted, and our review staff will go if they have a marketing application for a condition for which we have held a patient-focused drug development meeting. We may go to that and try to get a better understanding of what is meaningful to the patient, what their perspectives and experiences are. But after those initial FDA-led series, we have a lot of patient groups that are leading these patient-focused drug development meetings, and so they're coordinated by these patient groups. And FDA will participate in these as well. Many times we may provide opening remarks or closing remarks, or just attend these meetings to get a better understanding from the patients and their caregivers what is really meaningful to them in the context of dealing with their disease on a day-to-day basis. And then we have, patient engagement advisory committee. This is composed of patients, caregivers, and representatives of patients and organizations. Our FDA Center for Devices and Radiological Health actually established this to assure that the needs and experiences of patients are included in FDA's deliberations over review regulatory submissions. So I've gone over a few of these initiatives and how patients can be very helpful in our regulatory decision making. Thank
Great.
you.
I love all the opportunities that are available for FDA to hear the patient's voice. I think that's so incredibly important. Switching gears, so let's say once we have all the data, a sponsor has all the data, and they're thinking about bringing a drug through to approval for commercialization. What are the potential roadblocks that could impact approval? Jessary, do you want to start there?
Sure. So we touched upon this a little bit, right? If in the context of the clinical development program, we really identify an unreasonable risk to patients, that may result in a termination of a program, right? And that does not happen very often. We typically identify manners in which to risk mitigate. We may have risk evaluation and mitigation strategies that we can implement post-approval. But some of the issues that we see often, and Sandhya mentioned this, is comparability. So as product development ensues, manufacturers may modify their product and they may have gathered clinical trial data. They may have completed phase 2 or even phase 3 trials with a version of a product. And then as they try to get through more commercial scale manufacturing, they may need to modify that product somewhat. And that new version of the product, we need to be assured that that version is comparable to the product that actually was utilized in the clinical trials that generated the data to support the safety and effectiveness of the product. And so often that comparability assessment is very critical. And so that may be a huge roadblock if they cannot demonstrate comparability. It may mean that we may need a bridging clinical trial. It may mean that we may need a brand new phase 3 trial because the product is so different that we are not able to rely on the data that was collected previously. So these are some of the issues that that we tend to see. But again, I mentioned large scale manufacturing, that becomes an issue, and I'm sure Jennifer or others can speak a little more to the challenges that they face going from maybe academic centers that may have initiated the trials and they can manufacture this on a very small scale, patient by patient. But when you get to the marketing stage, you have to have product available to support marketing this and launching this and in the patient community. And if they have not, the sponsors have had challenges in large scale or commercial scale manufacturing, that's a big roadblock.
Sure.
And maybe piggyback on that too, if I can, real quick, Emily. Sorry, is just touching on that is, from the manufacturing, you also have to administer the product, right? And so finding centers, setting up centers that can administer a commercial product is also a challenge. So, we've conducted these clinical trials in highly specialized, usually academic centers. But patients are everywhere. And so do we need to have them all come to the centers that ran the trials? Can you set up new centers? Can they be set up in hometown locations that may not have any academic affiliation at all? And so all of those considerations can also be roadblocks to just kind of disseminating an approved product.
That's a great point. And it transitions nicely into, okay, so once a product's approved, what does success look like? But what are some of the roadblocks? Jennifer, can you speak to that?
Yeah, and I know we're coming on out of time, so I'll try to make this short. But I think ultimately, in the end, success truly is when the people who need the therapy have access to the therapy and can get it. That is truly what success looks like. But there's going to be roadblocks. We already touched upon earlier. There's logistical roadblocks. Transporting the therapy to where the center it needs to get to, setting up a network, getting people to the centers that can handle the gene therapy. I think there's also barriers around reimbursement. That's a big one and one that we often hear about. Gene therapies right now are expensive. And some of that comes to the complexities with the manufacturing that we talked about too. So, that's another roadblock that we need to keep working on and overcome. And I could keep going, but there are quite a few. But ultimately, success is we need to be able to figure out how do we get therapies to the people who need them.
And I'll just add one quick question. I realize we're going over, but Kim, can you talk about, this is such a great area where advocacy groups and patients and families can help. Can you touch on that before we close?
Yeah, absolutely. I think it starts at the very beginning that we've all kind of talked through ad nauseum, and it continues all the way through the end. Sometimes advocacy groups can even identify those physician partners or can equip their families with information packets about the disease, about the therapy, and work with the academic sites to help kind of develop those educational modules. And I know that that's been a new recent push is how do we educate everybody about the gene therapy, about the disease, and how to kind of collaborate and making sure that there's open dialogue all across the board.
Thank you, Kim. And thank you to all of our speakers today for this amazing insight on the various roadblocks to developing a treatment and potential solutions where patients and families can help. I also want to thank our audience for attending the session. We would love to hear your thoughts on this so that we can keep providing great content to you. So please, if you could fill out a brief 3-minute survey, you need to scan this QR code. So hold your camera up here. It'll take you to a screen on your phone where you can answer a quick 3-minute survey. Also know that the recording for the session will be available on patienteducation.asgct.org early next week. So feel free to share with friends or colleagues who couldn't make this. All previous Lunch and Learn sessions are available to watch on demand on the site, along with a whole library of Gene Therapy 101 and disease-specific content that is free for all to access and share. Note that the next Lunch and Learn will be held on April 27th, and will introduce the various kinds of delivery systems of non-viral vectors. Registration will be open on the ASGCT patient education site after this session, and we hope you'll join us again then. Thank you, everyone. Thank you to our panelists. Thanks for all who have attended, and I hope everyone has a wonderful day.
Thank you.
Thank you. Bye-bye.